Cloning strategies, gene cloning strategies. Okay, a clone refers to a group of organisms, cells, molecules, okay, or other objects arising from a single individual. Clone and colony are almost same. Okay, gene cloning strategies in relation to recombinant DNA technology broadly involve the following accepts. Okay, generation of desired DNA fragments, insertion of these fragments into a cloning vector. Okay, introduction of the vectors into host cell, selection or screening of the recipient cells for the recombinant DNA molecules. Okay, cloning of genes and screening strategies and the polymerase chain reaction for in vitro conditions, large quantities of DNA. Okay, gene cloning also involves the expression of genes, which is described under manipulation of gene expression in host cell. Okay, different cloning strategies is there. Okay, currently I explain shotgun experiment. Okay, shotgun sequencing method introduced by Sanger in 1977. Okay, first you take cosmic DNA. Okay, what is the cosmic DNA? It is the vector. Okay, cosmids are the vectors possessing the characteristics of both plasmid and bacteriophage lambda. Okay, cosmids can be constructed by adding a fragment of phase lambda DNA, including cos site, to plasmids. A foreign DNA, about 40 kilobase space, can be inserted into cosmid DNA. Okay, the recombinant DNA so formed can be packed as phages and injected into E. coli. Once inside the host cell, cosmids behave just like plasmids and replicate. Okay, the advantage with the cosmids is that they can carry larger fragments of foreign DNA compared to plasmid. Okay, this is cosmid DNA. Take the cosmic DNA. Okay, cosmic DNA, it is having two parts. There are uh, plasmid and phase lambda DNA. Okay, next step, sonication method. Okay, sonication. Sonication is the act of applying sound energy to agitate particles in a sample, which sample cosmic DNA. Ultrasonic frequencies less than 20 kHz. Okay, after sonication process will be completed, next random fragments will be come. Okay, it is random fragments. What is the random fragments? The starting DNA is okay, the starting DNA, it is a cosmic DNA, okay, is broken up randomly into many smaller pieces sort of in a shotgun experiment. Okay, with each of those pieces then sequenced individually when identical regions are identified, they are overlapped with one another. Okay, it is the random fragments. First step, you take cosmic DNA. Next, sonication. Okay, sonication means ultrasound energy, ultrasonic frequencies. Okay, will be applied. Next, this is random fragments will become. Okay, next step, you can add T4 polymerase enzyme. Okay, what is T4 polymerase means? It can be used DNA repairing process. Okay, it is a catalyzing agent. Catalyze is the incorporation of nucleotides into double stranded DNA in a 5' to 3' direction. It possesses a strong 3' to 5' exonuclease activity, but does not exhibit 5' to 3' exonuclease activity. Okay, T4 polymerase, it can be used catalyzing agent. Okay, it is the DNA repairing process. It is very useful enzyme, catalyzes the incorporation of nucleotides into double-stranded DNA in a 5' to 3' direction. It possesses a strong 3' to 5' exonuclease activity, but does not exhibit 5' to 3' exonuclease activity. Okay, next step, agarose gel electrophoresis technique. It can be used. Okay, these DNA fragments. Okay, the DNA samples are placed in the wells. Okay, this is wells of the gel surface, and the power supply is switched on as the DNA is a negatively charged group. Okay, DNA fragments move through the gel towards the positive electrodes. Okay, the rate of migration of DNA is dependent on the size and shape. In general, smaller linear fragments move faster than the larger ones will be slowly movement. Okay, hence gel electrophoresis technique will be completed. We can observe 3 kilobase pair and 1.5 kilobase pairs fragments will become. Okay, next purified these DNA fragments. Okay, you size selected fragments. Okay, this is blunt end ligation into sequencing vector. Okay, next step. Okay, this is DNA fragments. 3 kilobase per 1.5 kilobase per DNA fragments. Okay, size selected fragments. Next step, you take vector M13 vector. Okay, M13 vector is a single stranded DNA phase of E. coli, okay, or SMA1. Okay, inside the host cell, M13 synthesizes the complementary strand to form a double stranded DNA. Okay, for use as a vector is isolated, and a foreign DNA can be inserted on it. This is then return to the host cell as a plasmid. Single-stranded DNAs are recovered from the phase particles. Phase M13 is useful for sequencing DNA through Sanger's method. Okay. This is M13 vector. You can isolate the SMA1. Okay. This is the bacterial species. Okay. SMA1 means Serratia marcensis. Okay, it is the bacterial species. Next step, CIP. Okay, CIP means the DNA fragments sterilization sterilization of bacterial species. SMAI bacterial species. CIP means clean in place. Okay, these SMA bacterial species sterilization of DNA fragments. Okay, bacterial fragments. Okay, next step, size selected fragments. Okay, this is size selected fragments. So 3 kilobase per 1.5 kilobase per fragments. Next, these SMAI bacterial species fragments. Okay, both are insert into the one plasmid. Okay, through ligation process, it can be used ligation process. It can be used a ligase enzyme. Okay, after ligation process will be completed, these recombinant plasmids transform into the E. coli cells. Okay. Okay, look at here. M13 following ligation, the DNA is used to transform appropriate Escherichia coli host. Okay, are you understand? Transform into the E. coli host. Okay, laboratory conditions, you can maintain the laboratory conditions will be applied bacterial species. Okay, E. coli species. Recombinant plasmid transform into the E. coli species. Okay, these E. coli species grown in laboratory conditions. You pick the only good colonies. Okay, next step, colonies. These colonies are clones. Okay, next, recombinant clones. Okay, next step, PCR technique method, it can be applied. Okay, next, double stranded template or single stranded template will become. Okay, template means DNA replication process. Each strand of the duplex act as template uh, for synthesis of a new double helix. Okay, a molecular mold that shapes the structure or sequences of another molecule. Okay, template, just like 5 prime to 3 prime direction template, you see, observe, thymine adenine cytosine thymine, again thymine adenine cytosine thymine. Okay, just same repeated uh, nucleotide sequences is you can observe that type of sequence. Okay, okay, we will take system reference whether sequencing reading correct or not. Okay, Okay, the raw data are edited to remove vector and detail sequence of low quality and or computer assembled using current software. Okay, don't confuse. Shall I repeat? Okay, this is cosmic DNA. Okay, cosmic DNA, it is having two parts. These are phase lambda and plasmids. Okay, both are insert into the cosmic DNA. This is called cosmic DNA. Next step, sonication. Sonication means by using ultrasonic method, we cosmid vector, okay, will fragmented into different DNA particles, okay, that is called random fragments, okay. The starting DNA is broken up randomly into many small pieces, sort of in a shotgun experiment, with each of those pieces then sequenced individually when identical regions. Okay, this is random fragments. Okay, next you can observe. Okay, add the T4 polymerase. It can be used DNA fragments repairing process. Okay, it is the catalyst. Next step, agarose gel electroporation technique. Okay, you can look at here. This is a 3 kilo base pair, 1.5 kilo base pair DNA fragments. Okay, the size selected fragment typically in the size ranges 1.5 to 3 kilo base pairs. Okay, it can be applied power supply is switched on as the DNA is negatively charged groups. DNA fragments move through the gel towards the positive electrode. Okay, after migration process will be completed, you observe DNA fragments that is called 3 kilo base pair, 1.5 kilo base pair, size selected fragments. Okay, these are selected DNA fragments. Next. Next level, we can take M13 vector. Okay, this is M13 vector. We capture SMA1 species by using CIP, means we due to the sterilization process. Okay, with the help of restriction enzymes, we cut SMAI fragments, DNA fragments. Okay, next, these SMAI DNA fragments, next 3 kilo base pair, 1 kilo base pair, your already selected DNA fragments. Okay, both are insert into the one plasmid. It can be through, it can be used ligase enzyme through ligation process. Okay, will be completed. Insert into the plasmid, that is called recombinant plasmid. These recombinant plasmid transform into the E. coli cells. E. coli means bacterial species, Escherichia coliform bacterial species. Laboratory conditions, in vitro conditions, you maintain E. coli species, it will be developed, growth will be developed, colonies will be observed, will become. Okay, this is a colonies. This is the petri dish. Okay, this is agar medium. Okay, look at here, this is colonies. This is a recombinant clones. Okay, next step, PCR technique, it can be applied. Okay, double stranded templates and single stranded templates will become. Okay, templates sequences will be come. Okay, next cycling sequences. We will take system reference whether sequencing reading correct or not. Okay. Okay. Next, why its name shotgun experiment? In genetics, shotgun sequencing is a method used for sequencing random DNA strands, random fragments. Okay, sequencing random DNA strands. It is named by analogy with the rapidly expanding quasi-random short grouping of a shotgun. That is the reason it is named as uh, shotgun experiment. Okay. Quasi-random fragments mean short grouping, the great abundance of repetitive shotgun sequences. Okay, repetitive sequences. So it is a quasi-randoms. Okay. Okay. Look at here. Shotgun sequencing method consists of several independent procedures which are described in detail diagram. Okay, look at here. This is a diagram. The DNA is sonication to obtain fragments of the desired size. Next, the sonication material is end repaired using T4 DNA polymerase. Okay. Next, and fractionated by agarose gel electrophoresis technique. Okay. The size selected fragments. Okay. Typically in the size range 1.5 to 3 kilobase base. Next, purification process and inserted by blend 10 ligation into sequencing vector. Okay. Such as M13. Look at here. This is M13 vector. Next, following ligation process, the DNA is used to transform an appropriate Escherichia coli form bacteria. Okay, it is the host. Templates are sequenced using linear amplification processes and analyzed on automated DNA sequences. Okay, DNA sequences. The raw data are editing to remove vector and tile sequences of low quality and or computer assembled using current software. Okay, this is shotgun experiments. Okay, this is shotgun experiment. Any doubt?